Okay. Um, just really sort of quickly testing this. Okay. So basically, a couple of things happens. Couple of things happen as exercise actually increase. Exercise happens. Okay. Here's a nice little equation: mean arterial pressure equals to cardiac output times total peripheral resistance. Okay. And cardiac output equals to heart rate times stroke volume. Okay, that's very important to understand. Now, something cool that happens is that during exercise, sympathetic nervous system activity increases. Okay, if sympathetic nervous system activity increases, a couple of things tend to happen. Okay, let's start to go through it step by step. Okay, so what's really going to happen is that right here, in the central command, the higher brain center. Okay, we're going to get these descending fibers that go down to the respiratory center. And what they're going to do is, okay, they're going to go out. They're going to go out, and they're going to actually innervate the bronchial smooth muscle. Okay, the bronchial smooth muscle. As you can see, the bronchial smooth muscle cell, on their surface, they have the beta-2 adrenergic receptors that respond to epinephrine. Okay, and what actually happens is that that causes dilation. Okay, that causes dilation. Okay, so norepinephrine, epinephrine, whatever, that causes dilation. Okay, just sort of want to get you guys to understand that. Okay, that's that. Another thing that happens, okay, is that ventilation increases. Okay, so you're increasing ventilation. Okay, that should make sense. One more thing that happens is that you see this guy right here, okay, again, from the central command, they go down to the cardiovascular center and they go down to these alpha motor neurons. Before I talk about the cardiovascular center, let's go ahead and take a look at what's happening with the muscle. Because once you're exercising, what's really happening? It's the muscles that are sort of moving, right? The muscle's activated, the muscle's moving, all this stuff. Okay, so you have these descending fibers from the brain. They come down, they go to the level of the spinal cord, and they synapse on these motor neurons, specifically the alpha motor neurons. The alpha motor neuron activity here increases. If the alpha motor neuron activity increases, guess what's going to happen? There's going to be a lot of action potentials here. We're going to release a lot of acetylcholine on the muscles. Okay, so now you come over here. We're going to actually release a lot of acetylcholine on the muscles here. Okay, here's a muscle cell. Okay, if we release a lot of acetylcholine on the muscle cell, what's going to happen? What are we going to do? Let's come over here. Okay, so this nice little blow up diagram of what's actually happening over here at that point. What are we going to do? Okay, so here's our alpha motor neurons releasing acetylcholine. Very quickly, acetylcholine is going to actually do what? It's going to bind to these ionotropic receptors, which means what? Which means it's going to allow for sodium ions to come into the cell, causing an action potential, depolarization wave. That's going to travel on the sarcolemma, okay? The sarcoplasm, all right? And then it's going to travel, and it's going to go down the T-tubule. Once it goes down the T-tubule, there are these voltage-gated dihydropyridine receptors. These dihydropyridine receptors, they actually grab onto the ryanodine receptors on the sarcoplasmic reticulum, and they pull on them. They pull on them, and that exposes the sarcoplasmic reticulum now out into the cytoplasm. What is that going to do? That's going to start releasing calcium. When calcium gets released, what's going to happen? That's going to go ahead and bind to troponin. Once it binds to troponin, troponin moves tropomyosin aside. Once it moves tropomyosin aside, this orange protein here, it moves tropomyosin aside. Once it does that, what is that going to do? That's going to actually cause the myosin and actin cross bridge formation. Which when you form a cross bridge, what is that going to do? That's going to cause a power stroke. Okay, that's going to cause this actin and myosin sliding filament. Okay, that's that. But now we got to take a deeper look. Let's just zoom in on this sort of myosin here, and zoom in and see. The myosin head, it has actually two binding sites. It has a myosin ATPase, which binds ATP, and it has an actin binding site, which is the tips. The myosin ATPase site, it's very important. The myosin ATPase takes ATP, hydrolyzes it, and makes what? It makes an ADP and an inorganic phosphate. Where's the ADP going to go? The ADP it's very important. It's a stimulator for three different energy systems: the creatine phosphokinase, the glycolysis, and the electron transport chain. We'll get to see why. We have a whole different sort of whiteboard done on this before, but free ADP goes into the mitochondria. Okay, how does it go? It goes, from the outer membrane, it just enters through the pores. Okay, and then from the inner membrane to the intermembrane, it has to go through the adenine nucleotide translocase. It gets in there. Once it gets in there, this is very important, it's very crucial. Once it gets in there, it's going to go and interact with complex 5. Okay, it's going to open complex 5 and allow for the hydrogens to start flowing in. Okay, which allows for us to consume oxygen. Okay, and that's how we make a lot of ATP. Okay, from the electron transport chain. That's that. Now, it can also be an allosteric activator for phosphofructokinase, okay, which would increase the rate of glycolysis. If we increase the rate of glycolysis, what are we going to do? We're going to make a lot of pyruvate. The pyruvate can go into the electron transport chain, which will make a lot of ATP. But also another thing that can happen is that we can make a lot of lactic acid. How do we make lactic acid? When NADH dehydrogenase becomes fully saturated, because you know the rate of glycolysis is actually higher than the rate of mitochondrial respiration by 10 times. Those enzymes, their Vmax in the glycolysis is higher. Not going to get into that. Again, a whole other whiteboard. Where's lactic acid going to go? Lactic acid is going to go to a couple of places, actually. It can actually go out into the liver. Okay, it can go to the liver. And if you come over here, right, it can actually go out into the liver. Right, here's your liver. You can go out to the liver, and it's one of the stimulators for gluconeogenesis. Right, what am I going to do? If I do gluconeogenesis, what is that going to do? That's going to increase glucose, right? If I make more glucose, what is that going to do? That's going to increase glucose uptake by the muscle. Okay, that's great. Let's go over here. We're not done yet, okay? So that ADP that we made, you know ADP, it can be broken down to AMP and it can be broken down to adenosine. You know what adenosine does? Adenosine, we'll talk about it later, but adenosine can do a bunch of things. Okay, it can come out of the muscle and it can do a whole bunch of things. Okay, what's this guy over here? We talked about a lot of things here except this guy. We know the action potential has to end somehow, right? When the action potential ends, potassium goes out of the cell, right? Because we want to make the cell negative. So potassium has to leave the cell. What is that going to do? That's going to increase potassium. That's going to increase the potassium concentration out of the cell. What is that going to do? What is that going to do? That's going to go out. Okay, let's come over here. Let's actually talk about this. Potassium adenosine. These guys are called metabolites. Okay, they're metabolites. What are they going to do? They're going to come out here, all right? And you know how there's a blood vessel, right? That's supplying almost every single thing in your body, right? A blood vessel. Anyways, the blood vessel, what it's going to do, it's going to come out here. This blood vessel, something cool is going to happen. These potassiums, these adenosines, they're going to come here and they're going to cause a active hyperemia. What is active hyperemia? It's actually going to cause vasodilation of the muscle of the vessel. Why is that happening? Okay, let's actually explain it this way. You know, normally norepinephrine, right? Norepinephrine getting from the sympathetic drive. Normally, what's happening is that these guys will come and try and vasoconstrict these blood vessels. Okay, that's what normally happens. But these guys, these potassiums, these adenosines, they like to vasodilate. They cause a local myogenic mechanism, or what's actually called an active hyperemia, and they cause local vasodilation. That's great. That's amazing. Okay, that's what we want. What's going to happen if we vasodilate locally? We're going to decrease our total peripheral resistance. Okay, that's great. What is that going to do? That's going to increase our stroke volume. Why is it going to increase our stroke volume? If I decrease my total peripheral resistance, that's going to lead to what? If I decrease my total peripheral resistance, that's actually increasing my stroke volume, right? Why is that doing that? Right here, okay? Right here. And we'll talk about why. Because it's actually increasing venous return. Okay, it's actually increasing venous return. Okay, not important right now. Okay, but just imagine this: I'm decreasing my total peripheral resistance, which means what? I'm going to increase my mean arterial pressure. Okay. Sorry about that. I'm actually going to decrease my mean arterial pressure. Okay. That's active hyperemia. Okay. I'm just going to erase this actually. Okay, which is just to really fix this, sorry guys, active hyperemia leads to decreased total peripheral resistance, which means decreased mean arterial pressure. Okay, so right now we decrease our mean arterial pressure just from doing all this stuff. Okay, but we decreased it modestly. Okay, not that much. Okay, not by that much. But there's other things that can happen during exercise. Okay, other things that can happen. Remember these guys, these potassiums, these adenosines, they can do other things. These guys will come over here. There are special afferent fibers located on the muscle fiber. There's type 3 fibers and type 4 fibers. The type 4 fibers, they're called metaboreceptors. The type 3 fibers are called mechanoreceptors. They sense these mechanical sort of stretch in the fiber, okay? Both of these guys are part of the exercise pressure reflex. What they're going to do, they're going to get stimulated by the potassiums and the adenosines, okay? Specifically these guys, these metaboreceptors. What's that going to happen? These guys are going to go out. They're going to go send their afferent fibers into the medulla where the cardiovascular center is located. They're going to go out into the cardiovascular center. Once they're there, they're going to stimulate the cardiovascular center, which will do what? If you follow this pink sort of fiber here, okay, it'll actually increase sympathetic nervous system flow. Okay, it'll increase sympathetic nervous system flow. It'll come down here. It'll come down here. Boom, 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 boom. Where's it going to go? It's going to go out into the myocardium of the heart. It'll go out into the myocardium of the heart. Once it does that, what's going to happen? What's going to happen? It's going to increase stroke volume. It's going to increase contractility. It's going to increase force production. Okay, that's great for us. If we increase stroke volume, what am I doing? I'm increasing my Q. If I increase my Q, I'm increasing my mean arterial pressure. Okay, so I just increased my blood pressure. Okay, what if I go to heart rate? Okay, well, it's going to increase heart rate, right? Increase heart rate, increase Q, increase mean arterial pressure. But there's a little bit of a mechanism here. When sympathetic nervous system gets activated by the exercise pressure reflex, due to what? Increased metabolites out of the periphery that activate the afferent fibers that go onto the cardiovascular center in the medulla. Okay, what's going to happen? Norepinephrine is going to get released, but your norepinephrine actually binds to beta-1 adrenergic receptors. It binds to the beta-1 adrenergic receptor on the myocardium of the heart, and it causes what? Okay, it's actually on the myocardium and on the SA node. Okay, and it causes this increased stroke volume and this increased heart rate. Okay, great, fantastic. We'll get into the mechanism a little bit later. Okay, but that's the exercise pressure reflex. Okay. Now, I told you that sympathetic nervous system activity increases in general during exercise, okay? So what's happening? The higher brain centers, they're sending, again, fibers down, which would go what? Which would actually activate the sympathetic nervous system, right? These little thoracolumbar regions, okay? So it's activating the sympathetic nervous system, which means going where? It's going out to many different places. It goes out to the adrenal medulla. It goes out to the pancreas. It goes out to the blood vessels, the veins, and the what? The arteries, the arterioles specifically, right? Because the arterioles, you know, they're the highest resistant vessels in the body, right? So they go out into the arterioles and do what? They actually release what? I want to see if you guys remember. Yeah, so they actually release norepinephrine, right, on these alpha-1 adrenergic receptors now. Okay, so over here it was beta-1. Over here it's alpha-1. So they release alpha-1 adrenergic. They release norepinephrine on the alpha-1 adrenergic, which causes a vasoconstriction in the viscera. Why are we causing vasoconstriction in the viscera? Well, because, guys, remember, we're exercising, okay? What does your viscera consist of? Like, what do you not want blood flow going to as much when you're exercising? Like your kidneys, okay? Maybe a little bit like your spleen, or all these different sort of useless organs. We want to vasoconstrict them so that when we vasodilate it locally here, we can send that blood elsewhere. We can redistribute that blood. Okay, so actually constrict here. Blood goes out. Where does it go out? It actually goes out right there. It goes out right there. That's perfect. That's exactly what we want. That's exactly what we want. Okay, so once we do that, what else can happen? What else can happen? The sympathetic fibers, they can actually come out here. Okay, let me actually come over here and show it. Okay, so the sympathetic fibers actually come out here. Again, they release norepinephrine. So they release what? They release norepinephrine. Norepinephrine binds to the alpha-1 adrenergic receptors on the what? On these veins, on these venules. Okay, when it does that, it causes venoconstriction. Venoconstriction. Okay, so imagine this, it gets smaller. Okay, venoconstriction. What is that going to do? That's going to increase venous return. Okay, great. That's amazing. We're going to increase venous return. What is that going to do? That's going to increase my stroke volume. That's going to increase my cardiac output. That's going to increase my mean arterial pressure. What am I doing here? I'm increasing my cardiac output. Why am I increasing my cardiac output? Let's come over here. The reason why we increase cardiac output during exercise is to actually increase blood flow to the working muscles, the active muscles. Okay, but remember, as a consequence, blood pressure can get too high. So luckily, we always have a response to that, and we'll get to that response in a bit. What else is actually increasing venous return? The skeletal muscle pump, okay, and something else called deep breathing, right? When you're actually deep breathing, something cool is happening. Okay, here's your diaphragm. Okay, over here's your thoracic cavity pressure. Over here's your abdominal cavity pressure. When I'm actually breathing, okay, deep breathing. What am I doing? I'm actually compressing the veins here, right, and causing a lot of venous return back to the heart. Okay, so I'm actually increasing thoracic cavity pressure. okay? Sorry, I'm actually decreasing thoracic cavity pressure, and I'm actually increasing abdominal cavity pressure, which is going to cause what? Which is going to cause blood to actually go to the heart. It's going to go back to the heart, okay? That's called my respiratory pump, or deep breathing, okay? There's also the Valsalva maneuver, which does the exact opposite, okay? The exact opposite. It's going to cause increased thoracic cavity pressure, and it's going to actually decrease abdominal cavity pressure, which is going to actually compress the vessels, okay? So you're going to get less venous return to the heart, okay? Okay. And that's going to be that for a second. Okay. Sorry, where I left off, guys, is that you were increased venous return, and the whole point is if I increase venous return, I increase my end diastolic volume, and if I increase my end diastolic volume, I increase my stroke volume, which would increase my cardiac output and my mean arterial pressure. And we talked about why we want to increase mean arterial pressure. Sorry, why we want to increase cardiac output during exercise, which unfortunately we actually end up increasing mean arterial pressure as well. Okay, a couple of things can happen with the sympathetic outflow. The sympathetic outflow can actually go into a couple of places. We talked about the vessels. Now it's time to actually talk about the adrenal medulla and the pancreas. Okay, so you know this guy can actually go out to the adrenal medulla, right? If it goes out to the adrenal medulla, 1 second, guys. Let me just actually check something very crucial here. Yeah, okay. So if it goes out into the adrenal medulla, the adrenal medulla starts secreting a lot of epinephrine and norepinephrine. Okay, how does it do that, and why does it do that? Well, it releases 80% epinephrine, 20% norepinephrine. Okay, and it releases it into the blood. Once it goes out into the blood, okay, it can actually come over here. Both of them, they can come over here and actually stimulate the liver, right, and stimulate gluconeogenesis. If they increase gluconeogenesis, they increase glucose output. If they increase glucose output, a lot of glucose goes into the muscle and gets taken up. Okay, great, great. But I want you to remember that glucose output from the liver actually starts decreasing after 1 hour of exercise. Okay, and if that starts decreasing too much, that's not good. Okay, because that can lead to hypoglycemia. Okay, and again, remember the normal amount of glucose in the blood is around 5 millimoles. Okay, that's really good around that age. Okay, okay. Another thing that can happen is that it can go out to the pancreas. Once it goes out to the pancreas, something great happens. It goes out to the pancreas. The pancreas, it has these beta cells and these alpha cells. The beta cells secrete insulin. The alpha cells make glucagon. Okay, so that's the cool part. Norepinephrine inhibits the beta cells, so there's actually less insulin. And norepinephrine stimulates the alpha cells, so we actually have more glucagon. So glucagon increases in the blood, and insulin decreases in the blood. That's great. We want that because insulin, it's actually an anabolic hormone, and it wants to actually preserve things. We don't want that during exercise. During exercise, we want to break down things and use them as energy. Okay, perfect. So glucagon increases, insulin decreases, and norepi and epi increase in the blood due to the adrenal medulla. All of this was because of sympathetic nervous system activity increasing. Great. What is that going to do? Okay, well, I already said that the norepinephrine and epinephrine from the adrenal medulla stimulates gluconeogenesis at the liver. But you know what can also happen? Decreased insulin and increased glucagon can actually stimulate the liver as well, which increase glucose output. Another thing is that decreased insulin, increased glucagon, and increased norepinephrine and epinephrine from the adrenal medulla would actually stimulate the adipocytes, right? Our fat cells. You know what our fat cells store? They have a lot of triglycerides in them. Okay, so they actually have a lot of triglycerides in them. And they actually, if I come over here, they actually stimulate this guy. They stimulate hormone-sensitive lipase. If they stimulate hormone-sensitive lipase, what is going to happen? That's going to break down glycerol. Okay, it's going to break down into two things. Break down into glycerol and free fatty acids. The glycerol will actually go into the blood, okay, and make its way up to the liver, right? And it's a substrate for gluconeogenesis. We said that lactate in the muscle was also great for gluconeogenesis, and alanine, which is just a breakdown product of some amino acids, okay, after some exercise, okay, you break down that, and then you use that for gluconeogenesis. Regardless, here's the important thing. Free fatty acids. The free fatty acids, where can they go? Where can they go? They contribute to energy. They contribute to energy. Okay, they go and they bind to albumin in the blood. Okay, and then they go all the way to the muscle. In the muscle, they can be used to what? They can be used to actually contribute. They can be used to actually contribute to energy production or ATP production. Okay, so they can actually right here, I can put free fatty acids. Okay, why though? What is that? What is that going to do? They're going to go into the beta oxidation cycle. Okay, where they will be broken down. Okay, and make acetyl-CoA, and acetyl-CoA goes out into the mitochondria, goes into the electron transport chain. Sorry, Krebs cycle, then the electron transport chain, and then you make a hell of ATP, a hell of a lot of ATP. Okay, that's great for us. That's amazing. Okay, so what did we find out? The short version is that the increased um, the increase in sympathetic drive led to a lot of epinephrine and norepinephrine in the blood, which stimulated what? Which stimulated these liver and the adipose tissues. Okay, which led to them doing what? Which led to them actually activating the three systems of energy production. Okay, well, right, the ones that we all talked about. Okay, that is fantastic. That is great. That's what we wanted. Okay, we want to make more energy during exercise. Okay, we've talked about all of these mechanisms of how and why we're increasing cardiac output and why, as a result of that, mean arterial pressure increases. Now what we got to do is figure out, okay, if mean arterial pressure is increasing, how do I actually decrease it? Okay, how do I like bring it down a little? Let's talk about that. You know, on the carotid sinus, right? So here's your aorta, here's your left subclavian artery, here's your left common carotid artery, which is going to branch out into the internal and external carotid. At the bifurcation point of that, we're going to have this carotid sinus. Okay, that's called the carotid sinus. And on the carotid sinus, okay, we have these afferent fibers of the glossopharyngeal nerve. Okay, there's general visceral afferent fibers of the glossopharyngeal nerve that actually fuse together to make the Herring's nerve. The Herring's nerve activity increases if the mean arterial pressure is too high, because the pressure is actually pushing on these guys and it's actually stretching these receptors. Okay, these are stretch receptors, they're baroreceptors. Okay, and they actually sense that there's actually too high of a pressure here. And that's going to send a lot of signals down Herring's nerve, which goes to the cardiovascular center in the medulla. What that's going to do is it's going to activate the vagus nerve, okay, which is the main parasympathetic nerve, and it's going to actually decrease activity in the sympathetic nervous system. What's going to happen then? The sympathetic nervous system, let's following along in this purple over here, it's going to go out into the the SA node, the AV node, whatever. Okay, it's going to decrease the heart rate. Okay, why? Because the SA node activity is lowered. It's going to actually decrease contractility, right? Why? Because we're decreasing stroke volume. Okay, let's follow the parasympathetic activity. What is that doing? That's releasing acetylcholine on what? On this SA node, not the contractile cell. Okay, it does not affect contractility, the parasympathetic nervous system. It releases acetylcholine, which actually decreases the heart rate. Okay, that's great. All right, so now we know how the baroreceptor reflex works. Okay, the baroreceptor reflex and the exercise pressure reflex, they work in opposition to each other. Okay, now again, we talked about all these sort of factors that determine the changes and the extent of the exercise pressure reflex, and the amount of the sympathetic drive and the degree of vasodilation, right? And a coincident reduction of total peripheral resistance in the working muscles, right? And the baroreceptor reflex actually combat against excessive increases in blood pressure during exercise, right? It's a really protective response, okay? Because during exercise, a lot of crap happens, okay? And we gotta serve to sort of protect that. Okay, so the baroreceptor reflex is really a protective response. Okay, guys. Now I've mentioned that before, that the cardiovascular center from the exercise pressure reflex, it allows the cardiovascular center to come down here and actually increase contractility in the heart. Now let's go through all the mechanisms that are going through that, right? How do we actually increase contractility, okay? So I'm going to zoom in. I'm going to zoom in on this sort of section here, okay? And then we get to see how we're actually increasing contractility, okay? We've talked about it a little before, but now let's talk about it some more, okay? Okay, guys, so let's go ahead and actually talk a little very quickly about this, okay? So what I've gone ahead and done here is I zoomed in on what's happening right here, where norepinephrine gets released on the beta-1 adrenergic cell, okay, to actually increase cardiac contractility, okay, and increase stroke volume in return. What's happening here? So let's actually take a zoom in. So norepinephrine binds to the beta-1 adrenergic receptor. This beta-1 adrenergic receptor is a secondary messenger system, right? It's a metabotropic. Okay, it's a metabotropic receptor. Okay, and it binds right at the cytoplasmic end. It has G-stimulatory protein bound to it. The G-stimulatory protein gets rid of GDP and binds GTP, okay, which actually moves over, right, and activates another enzyme on this plasma membrane called the adenylyl cyclase. Adenylyl cyclase gets activated, okay, and it actually cleaves the GTP on the G-stimulatory protein and turns it back into GDP. So now it actually goes back, and now it goes back, and now it becomes this inactive G-stimulatory again. Okay, the adenylyl cyclase, now that it's active, it binds ATP, okay, and it turns it into cyclic AMP. Cyclic AMP goes and activates protein kinase A. Protein kinase A moves over into the cytoplasm. And you see this other channel here. This is a calcium channel, L-type calcium channel specifically. It phosphorylates the L-type calcium channel. So now there's an increased amount of calcium coming into the cell. Okay, now that there's an increased amount, because you know normally there's a lot of calcium coming in from the action potential regardless, but now we have even more calcium in the cell. Now that we have even more calcium in the cell, that's going to cause what? Even more calcium-induced calcium release, right? Which will bind to these ryanodine receptors, okay? And it'll do what? It'll cause even more calcium to come out of the sarcoplasmic reticulum. If there's even more calcium in the cytoplasm now, even more calcium in the cytoplasm now, what is that going to do? That's going to, one sec. If there's more calcium in the cytoplasm now, what is that going to do? That's going to bind more troponin. If it binds more troponin, more tropomyosin gets rolled aside. If more tropomyosin gets rolled aside, what are we going to do? More cross bridges being formed between the actin and the myosin, which means a lot more stroke volume, a lot more contractility, a lot more force production, a lot higher stroke volume. Okay, that's that. Okay, so that's going to actually complete the sort of integration of what's happening during exercise. Okay. Yeah.